So we have some people here who we haven't heard from or met yet yesterday. So I think if we have a short round of maybe very, very quick 10 seconds presentation of yourselves, like your names, where you're from, and maybe a line on what you're interested in, that would be great. I don't know how we can do this. I could pick people from what appears first in my screen, but maybe we could. Okay, so the first one here in my screen is Wilson Silva.
Hi.
Hi, Wilson.
Hi.
And I say where you're from and what you're interested in to everyone.
Yes, well, I'm professor at the University of São Paulo, and now I moved to Guarapuava, the city that I presented yesterday. And there we are building one co-facility for single-cell genomic analysis. Now we have the basic infrastructure to do that, and we start to training people to analyze the data from single cell. And it's a big challenge for me because it's everything that is new, but we have the support from the government from Paraná to maybe they will launch some grants for different projects, especially now for single cell genomics. And we just created one genomic network with 13 institutions from the Paraná state. And we are using the expertise from the research from the network to push some research in single cell genomes. And we are very excited to provide that platform, scientific platform, to help HCA Latin America to conduct some project.
Maybe we can already take note of this as a strength, because we have then a new institute in the south of Brazil, which is equipped, and you have a network with 13 institutions who would be willing to get together to discuss projects, and you might have a grant for single cell analysis also through this platform. So I think this can be called a strength that we have here, right? I don't know what Christine thinks, but I think this looks like a powerful something for us to start with.
Great
Okay.
start.
Yeah. That's really great. Cool. Next here in my screen is Dr. Enrique Hernandez.
Yes, hello everyone. I'm Enrique Hernandez Lemus. I'm a principal investigator and a program coordinator at the National Institute of Genomic Medicine in Mexico. It's one of Mexico's NIHs dedicated to genomics and its impact in healthcare. And I also run a lab, and my lab has been focused for some time on trying to dissect gene regulation patterns at the global scale networks in cancer, particularly breast cancer. And in the recent time, we have been interested in the role that the different infiltrates of immune cells within the tumors are playing in the overall tumor evolution. So we are interested in studying multicellular gene regulatory networks in breast cancer.
Thank you so much. And your institute is also interested in starting to work with single cell analysis?
Yes, we have been using 10x for some time, and we are interested in using it to start not only developing the computational and analytical skills, which is more on my side of the work, but also on perhaps establishing dedicated facilities for single cell.
I think we can also put that as a strength. That's Mexico also coming up with already technology installed, just like Wilson was saying, and computational analysis team that could also help. I think that's great. Great. Thank you very much, Dr. Enrique. Okay. Next in my screen is Daniela Robes.
Hi, everyone. So yes, I'm Daniela Robles. I'm a group leader at the National Autonomous University of Mexico, also called UNAM, in its Juriquilla campus in Querétaro. And I have never worked with single cell data, but I would like to start using it. We've been thinking about starting for some time, and we have already been talking to the Mexican distributors of 10x, for example, genomics to start, but then the pandemic came because we did start talking about this at the beginning of the year. We study at the moment a rare type of cancer called acral lentiginous melanoma. It's not been studied deeply because it's considered a rare type of melanoma in other parts of the world, but here in Mexico is the most common, as well as in other countries in Latin America, Africa, and Asia. So we have now a collaboration with the National Cancer Institute of Mexico to collect hundreds of samples from these patients and do genomics, transcriptomics, PDX models for this type of cancer and try to identify therapeutic targets, drivers, and all these analysis that we can do. But we have not started with single cell data. So I'm very excited to see if there's any way in which we can start collaborating. Also studying NAFLD and NASH. There were some talks yesterday about that. So we're also starting studies on that.
That's great. You have the cohorts, right? You have the patients then that you
we do. Yeah.
We already have a collaborator for you here who is Enrique Hernandez, who's also over there, and he has the machine. You have the cohorts. So I think that's already great.
That is true.
That is one possibility. Very good. Having the patients and the scientific question is also a very important strength. So I think that biological samples can also be considered a strength here in the group.
Thanks.
Next in my screen is Christine Wells. She's our co-chair here in this session. Christine, do you want to say? So Christine has a lot of experience with the HCA group, so I think she can tell us a little bit about that.
I'm Christine. I'm a stem cell biologist and a genome biologist in Melbourne, Australia. I think most of the experience I've had in consortia have been with consortia such as Phantom, actually, which Jay Shin is also very familiar with. And the great thing about consortia like Human Cell Atlas and Phantom, I think, is not just the capacity to participate in global science, but also the networks of scientists that you expose your trainees to, PhD students, and the capabilities that you can do as a group are much greater than the capabilities you could do on your own. So it's a really exciting time to do science and to be able to build on the networks locally as well. I'm very interested to hear what Latin America will do, how your individual countries will support you, and how we in the rest of the world can support you as well.
Thank you so much, Christine. Thank you. Next here is Jay. Jay, do you want to tell them a little bit about what you're doing there within the HCA context?
Yeah. Hello, everyone. I'm Jay Shin, and I'm currently in Weekend, Japan, Yokohama. I've been in HCA since the inception, so since 2016. So I've kind of understand the complexity of it all, but at the same time trying to simplify it here in Asia to motivate and kind of bring people and build communities here. So one of the things that I focus on is single-cell RNA sequencing, looking at gene regulation, and that's why I joined this breakout session. And I study a lot of stem cells, cell reprogramming mainly, and cell fate determinations. So trying to manipulate gene regulation in order to induce cell reprogramming using single-cell technologies. So with that, we do use 10x genomics quite a bit, but we also have access to other single-cell platforms such as iCell8, which is a microwell based. We also have the Fluidigm, which we don't use anymore. We have other custom compatible droplet systems called Nadia, and I'd be happy to share with you what are the advantages and disadvantages of different platforms. But 10x is somewhat of a useful tool because many other labs are using it. So it's becoming a lot easier to integrate and standardize. So it's becoming like the Illumina of single-cell technology, and that's not always a good thing either. But nevertheless, we do 10x genomics. One thing that I would also vouch for is to consider multiplexing samples, and I'd be happy to discuss more about how to do this because that will significantly reduce the costs of your operation. So yesterday I talked about this IDA project, and we multiplex PBMCs from 16 different individuals to a single run. So that reduces the cost to 1/16. So I think that is going to be quite powerful and useful for a place where funding can be limited. So the starting cost is high, but if you calculate the cost per cell and per sample, it can be quite cost effective. So that I'll stop there, but I'd be happy to kind of share some of these experiences and help in a way to be more efficient. Thanks.
Could we agree that this can also be considered a strength in the sense of having common interest with the Asian HCA in reducing costs and having protocols that have already been established and validated, right? I think that
absolutely.
Yeah.
So we have protocols that we share, protocols.io. We'll be happy to send you guys the link, how to multiplex, how much you should load, how to demultiplex, and things like that. So I'd be happy to share that.
Okay, Jay, that's really great. I think for groups which are just starting to work with the platforms, that's really important because usually we have only the very, code protocols from the companies, right? And optimization is something that takes a long time. So I think that would be very helpful. And I don't know if anyone has a comment on that. Maybe we saw that Dr. Enrique is already using this platform too. So maybe he would be interested in directly talking to about optimization. It's good. Okay, great. So here next to my screen, I have Ignacio, what's the
Wichmann.
Yes, a little bit complicated last name. Okay, so
hi everyone, my name is Ignacio Wichmann and I'm a PhD in medical sciences here in Chile. I work at the Pontificia Universidad Católica de Chile, and I just finished my PhD about a month ago. So my doctoral thesis was based on the transcriptomic changes and changes in stem cell biology and reprogramming of gastric stem cells into gastric cancer cells. So I was looking for transcription factors that could regulate the expression of long non-coding RNAs in this process, and how these changes could actually affect the balance of long non-coding RNAs and messenger RNAs that share microRNA binding. So I only did this from bulk RNA-seq and bulk microarray data, only public databases. And we got some really interesting results. And one of the questions that Vinicius actually asked me, because he was in my thesis committee, what would I think about doing single-cell RNA-seq for the this transformation and reprogramming of gastric stem cells into cancer cells. And I don't have any experience or funding, but I think that for people studying cancer, single cell analysis would be really interesting to see how these cells reprogram and where the origin of cancer lies, if it's within resident stem cells or other cells that become stem-like in the process of cancer trait acquisition. And now I'm applying to a postdoctoral grant where I'm going to study also the response of T cells and exhausted T cells to neoantigens. So that would also be a really interesting field to apply single cell transcriptomics on.
Mm-hmm. That's great. I think we have a lot of people here with interest in stem cell and cancer. So I think that's very, very nice to know. And do you have over there also cohorts? You work with data analysis, right?
Yep.
Okay.
That's just public data sets, and they have some cooperations with Vinicius Group at Universidad de Chile, and also with Elmer Fernandez in Córdoba, Argentina.
Okay, okay. Also, you're already working with Vinicius. Okay, I was gonna just say that. Okay, well, that's great. You already have a little hub there of people, of groups that are interested in single cell analysis.
Yep. And Carolina Bizama, who's also here. She's going to talk afterwards. But
okay. I think that's also an interesting strength that we have a group already formed in Chile that is already actively working with the analysis of data sets. I think that looks a bit like us here, right, Wilson? Because we have a group of people already working with data analysis. We haven't really generated our sets yet, but this is moving forward fast, I think, at the understanding data analysis. Okay, great, Ignacio. So yeah, next coming up is Carolina Bizama. I think you are colleagues, right?
Hello, everyone. First of all, congratulations for the initiative and invitation for this workshop. I am researcher. I am a doctor of science, and I am actually working as an assistant professor of the Department of Pathology in Pontificia Universidad Católica de Chile in School of Medicine. The School of Medicine actually have a biobank of tissue and blood fluids, and I actually I am a principal researcher of a Fondasit, project of the government of Chile, focus on the use of organoid platform, has a model of tumor biology in gastrointestinal cancers, and has a platform to predict the response of chemotherapy and immunotherapy in gastrointestinal cancer. And I have not experience in single cells analysis, but I would like so much can be characterized my organoids, in specific the plasticity of the stem cell inside of the organoid. And I would like so much contribute and collaborate with the samples, the organoid sample or tissue samples that are necessary to complete the research.
Thank you, Carolina. That's great. So can we also add this to our strengths that they are working with organoids. They also have a biobank, and they have this very streamlined. I think this is great. And there is also this connection with the HCA organoid working group. They have a lot of information on that. So I think that is an interface that we can also explore and consider it a strength, plus the biobank, because that's really great. Thank you. Thank you so much. Next year, yeah, we'll talk more later. Everyone, keep in mind what we are discussing, and afterwards we're gonna come back to this to see what challenges are for us to be working together with these groups, and how we could get research funding and maybe some small hubs within this breakout session that could already start discussing some collaboration. So take notes of what you're listening, and afterwards we are going to come back to this. Next here, thank you so much, Carolina. Here next to my screen is Marilene Lopes. Hi.
Hi, everyone. So first of all, I'd like to thank the organization for this opportunity to share with you and discuss with you a little bit about our work. So I'm Associate Professor of Institute of Biomedical Sciences, University of São Paulo. And I'm very interested in understand the biology of stem cells in different contexts. In glioblastoma stem cells and also pluripotent stem cells, we have potential candidate to modulate both biology, I mean biology of stem cells and also glioblastoma stem cells. So we work with PDX and also patient samples. We have a collaboration with AC Camargo Cancer Center in São Paulo. So we work with patient-derived xenografts and also patient samples. We have this specific protein is a cell membrane protein, and we know that this protein is different levels of this protein expressed in the glioblastoma cells. So we have this heterogeneous expression of this protein in glioblastoma. So both approaches, I mean, glioblastoma stem cells and also pluripotent stem cells are very heterogeneous. That's why I'm very interested in implementing my laboratory, the single cell analysis, to understand a little bit more about the contribution of our potential candidates in the biology of the cells.
That's great. I think we already have a clear common interest with Wilson, who is also working with glioblastoma. I don't know if you're familiar with that. And he has a great center for single cell analysis, right, that he was just describing. So I think that that can be also noted. Thank you. Next, we have here another USP colleague, right? João Paulo Fabi is in my screen here.
Hello, everyone. My name is João Paulo Fabi, but you can call me JP. I think it's better and it's easier. So I work at the University of São Paulo. I am an assistant professor, and I work in the nutrition and food department. So I am interested, and I'm working with the dietary fiber and the fermentation and the results of the short chain fat acids in the biogenesis of colon cancer and the inhibition of colon cancer. And I work also with organoids, so with colon organoids. And of course, I don't have an experience with single cell RNA. I am a colleague of Elder Nakaya, so he said that, of course, we could join some forces to try to make some findings. So that's the reason that I am here.
Thank you so much, João Paulo. I remember you from some of the initiatives from last year, right?
Yeah.
And you're interested in that. And I think this is like building on that discussion from last year. So yes, we think that we are gonna come up.
Yeah, definitely. And I am a part of a CPD from FAPESP, that it's called FORC, that it's Food Research Center. So we are trying to join all the force we could do it for it. Thanks, everyone.
Thank
JP,
you.
I think it's a really great strength if you can bring different modalities to the analysis of single cells. So transcriptomics or DNA is one modality, but being able to look at metabolomics across particularly in situ sections would be hugely interesting as well.
Yeah, we have a facility for metabolomics and proteomics, but we don't have the transcriptome one. And I am the only one that is working with RNA-seq in my group. So I was trying to see how we could do it, starting from organoids and afterwards trying to get some patients, but I don't have the cohort. Patricia knows that, but of course we could join something and someone from biobanks too.
Yeah, they have, you have the hypothesis, right? I remember you had hypothesis and he needs the samples, but they are, that's great, great, yeah.
Thank you.
Okay, so I don't know if everybody saw in the chat that Jay Shin shared with us a link with protocols. That's great. Thank you very much, Jay. Next in my screen is Mariana Boroni. Hi, nice to see you again today.
Hi, Patricia. Hi, everyone. So I am Mariana Boroni. You can also call me Mari if it's easier. I am the group leader of the Bioinformatics Lab in the Brazilian National Cancer Institute. I am also one of the CZI grantees. I am really interested in study the tumor microenvironment. We are very interested in the plasticity of some cells in this microenvironment, especially the myeloid derived ones. Regarding the resources we can offer, here in the Institute, we have a sequencing facility. We also have a computational platform that can be available. We are trying to acquire single cell RNA-seq platform. And we also have a tumor biobank here. So we can have several samples. Right now they're frozen, but we can also manage to run new, to get new samples, fresh ones. So.
Mari, do you have the support from INCA? What does INCA think about this? Has this been discussed? This institute, I'm just asking her, is a very important cancer institute in Brazil. They have a lot of samples and a lot of very strong research groups. Have they been discussing this internally?
Well, just in the research, but not like in the whole hospital, but I'm sure they were very glad with the CZI grant, so I'm sure they will be very happy and very happy to help as well. So I don't think this is going to be a problem.
We were all very happy with your CZI grant. We were all very proud. Yeah, it was a very big, I don't know, like a very big, how do you say? I forgot the word, something that, anyway, we're all very happy, very, very proud of you guys for having got that money.
We are very glad, and we are willing and to help you guys as well, and also to fortify these new hubs that I'm sure it's gonna.
And don't forget that now all the eyes are on you, and you have to do a great job because you are going to be the reference from Brazil working with this.
Oh my gosh, no pressure on me.
Yes. But we are very, very sure that you guys are going to do a great job. But anyway, we're all like hoping that everything goes as good as possible. Thank you. We'll get back to you. So this is a great strength to have people from the INCA in the group, because they're a big cancer center in Brazil. They have a lot of resources, a lot of samples, a lot of patients, clinical trials, right? And a lot of drug response also cohorts, right? So it's great. Thank you. Next here in my screen is Dr. Masako Suzuki. Hi.
Hello. Hi, my name is Masako Suzuki from Albert Einstein College of Medicine in the Bronx, New York. And I am interested in the prenatal environment, how that affects to the health, the offspring later in the life. Especially the Bronx, we have so many, the Hispanic, Latino, the population in here. So that is why I'd like to join this team to see, like the Latino, the Latin American, the researcher is working how, working on the single cell, the studies. That is why I joined. Mucho gusto.
That's really great. I was wondering because we didn't have many people coming from the US, right?
Yeah.
And now I can understand why you joined. And that's such a great idea. I think this is a great strength for us to be able to collaborate with a group from the US with a population that could be similar to the original countries from Latin America. That's really, really great. I hope this is, I'm sure this will work out. Thank you. Thank you for joining us. Let me see if we have someone else that has, and yeah, Vinicius.
Hi, Patricia. I am Vinicius Maracaja Coutinho. I'm Brazilian, but I'm currently located in Chile. I am a group leader at the University of Chile, and also I am an associate professor of the Advanced Center for Chronic Disease, which is one of the center of excellence that we have here in Chile. This is a center it's co-hosted at the University of Chile and the Catholic University of Chile. And in this center, we follow up a cohort of 10,000 people from a particular small town in the south of Chile. And also through ACTIS is the acronym of this center, we can have access to the both University of Chile and Catholic University biobanks. Also, I am a permanent faculty of the PhD program on bioinformatics from the Federal University of Rio Grande do Norte in Brazil. And I am director of a postgraduate course on bioinformatics and computational biology here in Chile. My research interests are related to gene regulation and also development of tools for data analysis. And now we are trying to characterize at a single cell level the different non-coding RNAs. And another field of research that we are interested is the epitranscriptomics. And we would like to go to try to how can I say, to put together, I don't know, the nanopore sequencing with 10x genomics in order to obtain direct RNA-seq, and then perform deeper analysis in order to identify modified sites in these transcripts. Right now, we together with other Center of Excellence here in Chile, we applied for a funding from the national government related to the acquisition of equipment. The results we are expecting last week, but we are not, I don't know, I'm here with another screen updating every time because we can have maybe a Chromium here in Chile, and the result I believe that we will have it today. Related to networks.
If you do, don't forget to email us, because we are all expecting that too.
Of course. Currently, I am a very close collaborator of Elder Nakaya, and also different groups in Brazil, Argentina, and Peru. And all these groups, they are already starting or interested in perform single cell transcriptomics or genomics, integrate into their research lines. And also, just to finish, I am also connected with Cabana. That's a national, it's a network led by EBI, the European Bioinformatics Institute, with the aim of, I don't know, to improve the workforce on bioinformatics in Latin America. Together with them, at the beginning of this year, we're starting to organize a hands-on workshop on single-cell data analysis. The idea was to have it here at the south of Chile, in the Chilean Patagonia, but the COVID-19 stopped us. But we are still planning to, as soon as we have this situation solved, the idea was to have this hands-on course here in Chile, with people specifically from Latin America who are doing something on the field, of course, with some speakers from abroad, from EBI and other centers. This is it. Thank you.
This is really great. I don't know if Jay and Christine would like to comment on this, because a workshop on data analysis in Chile for South American researchers. I think Wilson is probably involved with that too, right?
So, I mean, I think it's one of the most straightforward things that you can do in a collaborative sense is share data and share code. I would encourage you, if you're going to be sharing data, to look at the data portal for the HCA, you know, making sure that you've got the metadata associated with the experiments, so that the phenotype of the tissues and samples that you've collected, and also the experimental platforms that it's been run on, are really well articulated. I'm not sure if the data portal are accepting the long read sequencing at the moment, but I certainly know that there is some long read RNA that's being paired with single cell data in the kind of nanopore community.
Yes.
So please get in touch if you aren't able to get hold of any of that.
Oh, great.
I think that the organization of the data analysis groups, we have in mind having this core group, right, Wilson, that could be supporting the whole network.
Yes, I think it's a great idea, maybe the next semester, 2021, to organize one workshop, like Latin America single cell workshop with hands-on, maybe 2 or 3 days for lecture, to training theory about the equipment, the method, the protocol, and the rest of the week or to do the hands-on in different spots, like in Chile, in São Paulo, Paraná, Mexico. In the same time, we can use the public data. Maybe we can discuss one biological question and take the data, the public data, to do the hands-on. I think it's for training people is very, 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 very interesting. We have the experience here in summer courses, work very well.
Specifically for what Vinicius is proposing, which is data analysis, you can
Exactly.
do it even if you're not physically together, right?
Exactly.
Exactly, and we are discussing because the idea of having this hands-on workshop physically, it was to put together people and they stay there for a week and have all that connections. But at the current situation, I think we are discussing with people from EBI to change it to an online course, and I believe it would be great to have you on it, Wilson. The idea is to put together people from the goal is the same, because we are started that with Cabana with this idea to put together people from Latin America and prioritizing people from here, 10 people from abroad.
That's great.
I think that's a pretty good point for us to already highlight as a strength, which is data analysis groups organizing themselves for data sharing and protocols for data analysis. That's a really great news, Vinicius. And if you can have most of the groups involved, especially if it's going to be online, then you can really invite a lot of these South American groups to join, right? That's great. Mari also has a...
I would like to say that I can also help on that.
Yeah, I was going to just say that Mariana has a very good group there too. So yeah, thank you, Mariana. Yeah.
Can I encourage you to reach out to Timothy Tickle, who's with the Human Cell Atlas data portal. And I think he would be really interested in participating in something like this. The data portal group are very, very keen to support easy data use across the HCA network. And they have They have a good team, not just with the husbandry of the data, but also they have data wranglers to help you get the data out in the formats you want as well. So they're interested in partnering with communities to understand what it is that what your use cases for the data are.
Yep, you have one here today, so
okay.
Any questions? Yeah, sorry, I
was just gonna say Enrique's here.
Yeah, I have a very unstable connection today, sorry. That's why I have my cam turned off.
That's great.
Enrique was in the talk yesterday, remember Vinicius? So he's one of the data wranglers.
That's great.
And he's a very nice person, so.
Thank you. Yeah, so if you have any question, either about metadata or whatever, like point in the how to start a contribution or whatever, just ask me, can help you. So, or just email the data wrangler team at wrangler-data at, let me check that again. I don't just use it too much, so.
You can send it to us in the chat. I think that's good.
Yep.
Yeah.
I will send it 1 second. Okay.
That's great.
So good to have one of them here today. I think that's one of the things that can, data analysis is what we can do, right? First, and it's very important to have it going before we have actually data from biology. So I think it's great what you guys are doing. All right, so let me see. Next here in my screen is Hernandez Carvalho. Hi.
Hi, hello. I am from Campinas, São Paulo. I teach cell biology in the University of Campinas, or better known as Unicamp. And I've been working with the prostate gland, mostly using animal models. And I've been doing some ChIP-seq, RNA-seq, ATAC-seq, like to answer specific questions, but never work with single cells. And I believe this is a good opportunity to change the gears in my lab, just to go into human samples and perhaps doing single cell analysis. I'm very enthusiastic of this approach, so I'm very excited.
That's great, and it's great to have somebody from Unicamp here, right? You can also stimulate your colleagues over there to join us, right? Because
sure.
Is huge, right? So we're trying to reach out for these big centers in Brazil, and great to have you here. You have a lot of experience in RNA-seq, ATAC-seq, and other methodologies that you can also share with us. Thank you so much. Next here in my screen is Gustavo. Hi, Gustavo.
Hey, hello everyone. I'm Gustavo Amarante Mendes. I am a professor and the vice director of the Institute of Biomedical Science, where the same as Marilene and Lucio are operating. I am somewhere in between cell biology and immunology. So I work quite a bit in the past with leukemia. Now I'm moving to melanoma. I'm interested in also in, from the immunology point of view, the macrophages dynamics and CD8 T cell differentiation, tumor microenvironment. I'm developing organoids as well, like artificial skin, mostly with, we're working with mice. Also, we have connections to different hospitals, including the Einstein Hospital where Patty works. So I have a personal, I have never worked with single cell. I'd like to add that to the projects, to the approach we are using in the lab. But also as the vice director of the institute, we want to be one of the reference institute in Brazil. We already have huge facilities where from proteomics, genomics, flow cytometry, imaging, that we offer service to the whole community. Again, particularly in the city and state of São Paulo, but also people from all over the country come to us to develop some of their experiments. So we would like to adapt the single cell and be one of the references in the area. Thank you.
Thank you so much, Gustavo. Gustavo is very, very experienced in all these topics and has been working with cell biology and molecular biology for the longest time, also immunology, and the infrastructure of University of São Paulo, and specifically of the Biosciences Institute, is very, very useful, important, really essential to us. So thank you, Gustavo, for joining us and pointing that out. It's very important to have your institute like committed to this, right? I know the facilities are great, so we will be using them.
Thank you.
Next, we have another colleague from University of São Paulo, is Tatiana Malta.
Hi, everyone. So I'm current and young investigator. I'm a grantee of one of these grants from FAPESP to help young people start their own lab. So I started my group 1 year ago at School of Pharmaceutical Sciences here in Ribeirão Preto. I have some experience in data analysis. I worked in a couple of projects in the context of the Cancer Genome Atlas, or TCGA. And my main goal right now is studying stemness in cancer. So that's part of my young investigator grant. But we are in this time that we notice that we acknowledge that the single cell data can improve our work. And we are very interested in studying the tumor microenvironment right now, specifically in longitudinal samples from gliomas, primaries, and recurrences. And I have a student that has been working with single cell public data, but we realized that most of the work that has been published focused on the tumor cells, and we are kind of more interested in the non-tumor cells or the immune infiltration. So I'm interested in generate some data, and I have access to tumor banks both here in Ribeirão Preto and also in the US. And I love to collaborate with people here. I noticed that they might have some interests in common.
Mm-hmm. Thank you so much, Tatiana. Yeah, a lot of people here talked about nervous systems cancer. And so, yeah, I'm sure. Plus to have a group of people from the University of São Paulo who are engaged, that's really important too. Thank you. Thank you.
Yeah, also actually, I also have a collaboration where we are interested in studying the inflammatory aspects of the sickle cell disease. So I also have access with people here at the blood center. And actually we are right now trying to get funding to support this project. Yeah, so.
Mm-hmm. A biobank, okay, that's great. I think that'll be good for us to talk a little bit about biobanks and sample sharing in a bit. We have, I think, 2 more people who haven't spoken. So I have Dalia Ramirez. Hi.
Hi, everyone.
Hi,
Dalia is also from Mexico, right?
Yes. Hello, everyone. My name is Dalia Ramirez. I am an associate researcher in Mexico. I am currently working on acute lymphoblastic leukemia, which is the main cause of death worldwide in pediatric patients due to malignant neoplasms. We perform cytomic of the patient to try to identify leukemia initiating cells and the accompaniment of the tumor by immune cells and how they are remodeled by the tumor microenvironment. The reason of being here is to learn about a little more about the big data analysis and other approaches to address the pathology.
Have you in your do you work with patients, right? You have patient samples, right? Okay, that's
bone marrow samples.
Thank you so much, Dalia. And who hasn't spoken? Joaquin, right? Joaquin Garat from Uruguay.
Hi, I am from Montevideo, Uruguay, and I am in my last stages of master's degree in the genomics department. And well, our group is interested and has experience in gene regulation analysis, such as transcriptomics and translatomics. And well, as a part of my thesis, we propose to start analyzing single-cell RNA-seq data in order to analyze ribosomal proteins, mRNA levels in single neurons. Part of these results were presented by Pablo yesterday. Perhaps you have listened to them.
Right? Yes.
Yes. And well, I am currently learning how to analyze correctly this data, and I'm willing to learn more and apply these things to more projects.
Great. You guys have a lot of experience with gene regulation, and I talked a little bit with Pablo yesterday. He said you would be joining us from Uruguay.
Yeah.
Yeah, it's great. We would like really to have a person from each one of the Latin American countries in each group, so we could interact and really maybe get together on something already. Thank you.
Okay.
Thank you for being here. I have also here Mauro Castro. Mauro is also from Brazil, but from from São Paulo State. Let's see if he's here. Hi, Mauro. Oh, we cannot hear you.
Mauro is from Federal University of Paraná.
Ah, from Paraná. We can hear you now. No, I can hear you very, very, very low. Can you put your volume up?
A little better now?
Little better.
Sorry, I in the math.
We cannot really hear you. Wilson, do you want to introduce him?
Can you try without the phone?
Hello, can you hear it better now?
No. Now it's some very high background noise.
Let me try to log off and log in again.
Okay, but can you hear us well? You can hear us.
Perfect.
Okay. So we wait for you to join later.
Okay, thank you.
I have another person here that hasn't talked. Eduardo.
Hello.
Hello.
Camera.
Hi.
It's Eduardo. I'm from Brazil.
Okay.
Universidade Federal de Santa Catarina. Maybe the only one from Santa Catarina here.
For sure, yes.
I have been working with single-cell genomics since 2015, when the data sets have only about 50 cells. And now we have this explosion in cell numbers. I did pretty much all my postdoctoral training at George Daly Lab at Harvard Medical School, and I have access to these technologies and analytical tools since they were in their early stages. So I'm very much experienced in that. I returned to Brazil by the end of 2018 and started my lab here at the Universidade Federal de Santa Catarina by the end of 2019, like November or so. And my lab focused mostly on developing new methods for analyzing single cell data. I'm originally a computer scientist, which migrated to biology over the years. And I have experience in every stage of experimental design, sample collection, and especially develop new algorithms that can help to address new questions. And in my lab, we do a lot of methods development and also experimental work in cancer and stem cells, especially trying to learn how the immune system develops from hematopoietic stem cells in the bone marrow, and then try to engineer induced pluripotent stem cells to create immune cells for cancer immunotherapy. That's one of the research lines in my lab right now. Since I started, I have been applying to several grants, international and national ones, to get started in some projects that we are about to start in the lab. So basically, that's it. I have a lot of experience with methods development and analysis of single cell data in cancer, stem cells, and any other tissue that might be of interest.
It's really great to hear from you. I was wondering why we had missed you last year when we were doing the workshops, because we were looking for people that had experience. But I suppose because you were not here, right? You just came back.
Yeah, I just came back, and also I think I'm a little bit shy, so I was trying to
avoid us. Okay. Too bad. Now I cannot avoid this anymore because now we know where you are.
Yeah, but actually I started last year, so I'm only now trying to build connections here in Brazil because I spent like 4 years abroad, 4 or 5 years abroad, and now I'm hopeful to connect to people here in Brazil and hopefully do some exciting science together.
As you can see, we have quite a few people who talked here who are really looking forward to building a group to work with single cell analysis. So you're most welcome. It's great to know that you are in Santa Catarina because we hardly ever hear from Santa Catarina, right? In our
I hope to change that.
Exactly.
To change that a little bit.
Yes, you're also near Wilson and his institute in Paraná. So that's also great. I really hope you guys are gonna get connected and start working together.
Yeah. Awesome. Sounds great.
That's
nice to meet you, Wilson.
Yeah, that was a very great surprise to meet you here today. Thank you.
Let's talk about that.
I appreciate that.
And
so the IPS models of blood development are really fascinating, I think. And there's some interesting parallels coming out from a few people speaking about the role of the immune system and inflammatory cells in different tumor microenvironments. And I think being able to model that from a pluripotent stem cell is really powerful because you've got opportunities of adding some of the lineage tracing tools and some of the reporter tools to look at immune tumor interactions that are hard to introduce to the primary cell. So that's really exciting technology.
That's great. Eduardo, are you setting up also the wet lab research or data analysis? Are you gonna be doing data?
Yes, my hope is that my lab is gonna do both the wet lab and the dry lab research. And I have been applying for grants to fund the dry lab aspects of the work because working with pluripotent stem cells is quite expensive. And here at Universidade Federal de Santa Catarina, I don't know anybody who actually does that. So I have connections with Lígia Pereira. She's in another session, and she's providing me some very valuable feedback to set up pluripotent stem cell lab here in the south, and let's see how it goes. But most of the work that we are doing in the lab right now, it's the dry lab stuff regarding novel development of new algorithms and reanalyzing data to generate hypotheses for when the money comes from the wet lab stuff. we have everything in place to get started on that.
That's great. That's great to hear. Meanwhile, if you can join with people who are doing getting together for data analysis, that's going to be just it's fantastic. Okay.
Yep.
We have one more person who recently joined us here is Catherine.
Hello, everybody.
Salut, Catherine.
I'm talking with you from France. I work in a commercial platform focused on NGS. And we recently, I'm in charge of making the validation for CE marking software for data analysis in cancer. And we are trying to develop for our customers new NGS techniques from wet lab to dry lab and data interpretation in a regulated context. So for delivery of clinical results. And I have a great interest in knowing what's going on in single cell and generally interpretation of microenvironment in cancer biology.
I think that if you can help us use different technologies or complementary technologies to what we're doing, I think that would be really great to have you around. So feel free to give your opinion.
My pleasure.
Yes, please share that with us. I think that with Catherine, we have heard from anyone. Is there anyone there that hasn't been contacted?
I think I managed to come back again.
Maud is back. Okay.
Okay, thank you. Sorry for my internet connection. I hope the connection is better now. Can you hear me? Okay.
Yeah.
So first of all, I would like to congratulate the initiative to bring this this meeting to Latin America. It's great, yeah, and for us that are really keen on data analysis, this is even more great. Yeah. My name is Mauro Castro. I am professor at UFPR University at Paraná. And I am a group leader in the area of bioinformatics. I coordinate a brand new PhD course that should start next month to contribute with human resources in the area, which I think is great for our state here. This is the first PhD course in the south of Brazil, dedicated to bioinformatics. And we are very enthusiastic in terms of community to bring people together and to dedicate efforts to assess high quality and high standard data sets produced by consortiums. We have close collaborations with several groups here in Brazil and in the United States, in Canada, in UK, in Holland. And so we personally, my group is dedicated to develop new methods in the area of data analysis, focusing on cancer research. One of the key aspects of my group is to try to find master regulators in cancer, master regulators and modulators in cancer, using what we call global coherent data sets. For example, when you, for the same set of samples, you have genome, epigenome, transcriptomes, ATAC-seq data, methylation data, and to integrate all these multiple levels of information for the same set of samples, which it's really challenging, not just for us, but for all the collaborators that I have the experience to work with. Single cell analysis bring another level of complexity, turn lives much more interesting, but much more challenged. So
have you tried that yet?
We currently have some methods that we are trying to migrate from bulk tissue to single cell, but they are not yet available for community.
Mm-hmm.
We have some collaboration with Penn State University at the United States, with some very early initiative to try to transform some of our methods to query master regulators in single cell data sets. But as I said, this is very mature, very early, but we are very enthusiastic in doing this bridge between bulk tissue and single cell.
That's really great to have you also from Paraná. I think that if we think about Brazil, Brazil incorporates many different countries because we're so big. So it's great to have people with contacts in the north and in the center and in the south. Thank you. Thank you so much for joining us. I think we got to hear everybody, and we have a very good idea of what we have within this break session group to discuss some specific points of what we want to bring to the rest of the group, to the big group. When we think about how we could best collaborate with a human cell atlas, we have some shared interests here, but it's a group that is working with cancer and stem cells. In this context, any opinions about our specific strengths on that? I heard about some rare kind of cancer that hasn't been studied, and organoid models too.
Yes, so I could start. Indeed, we think this is a strength of maybe something we could study. Actually, I've been talking to Mari, who's also here, and Patricia, who's also asked to be in this meeting, but she was put in the inherited diseases one. But we've been talking about this for some time now, and I think, or we think, it's a good thing that we could, let's say, direct from this area of the world also with Asian countries, because it's also a very important disease over there. So we've been collecting samples from Brazilian patients and Mexican patients and doing many different studies. We think this is a strength because we have been able to also get funding internationally from Wellcome Trust, from Medical Research Council, also nationally, because it's actually not very well studied, this disease, and I am not aware of any, for example, transcriptomic data sets, even bulk, that have been published from this. So I would think it's a good, at least one of the things we could suggest could be focused on that. And I know there's also interest from other people in this. Mm-hmm. And also, I've been writing with everyone as my job as secretary here, what everyone has been saying, and I also heard another researcher, I'm looking here through my notes, that also mentioned melanoma, Gustavo. Mm-hmm. All right, so could also be interesting for him as well.
And also Mari from Inca, that could look into this specific cancer types that are more rare and possibly more common in Brazil and Mexico, and that could be also assessed, right?
Yeah.
If we think about that, and the biobanks that I wanted to get back to, because we have quite a few groups here that mentioned they have biological samples. Before we talk about that, just to wrap up, that one big strength that we saw in this group is that we have a lot of people engaged in data analysis. So I think that for data analysis, we have a lot of already consistency in what's being done. Different groups, they share a lot in common about how they want to proceed with that. So that we can already say that's a great strength that we have already been established, right? And considering then the biology, we may consider that we have rare samples. So samples from rare cancer types, for example, that could be studied in different ways. And that would be more typical or found in Brazil or Mexico, for example. And if we don't think about the other, this specific cancer types, we also have a lot of resources considering biobanks in general, right? We have a lot of samples that should include a lot of genetic diversity. What do you think about that?
Maybe I just want to comment that biobanks are great, and I think Orit from MIT mentioned this yesterday at the technology standard working group, is you can use nuclear sequencing, single nuclear sequencing, and that really allows you to extract the nucleus and profile the gene expression without dissociation. So as long as the tissues are well preserved, you should be able to do single nuclear sequencing. And although I think you do have to be aware that there are discrepancies between dissociated approach and the nuclear approach. I think she already also mentioned, for instance, that infiltrated immune cells will be very different. So if you're studying T cells, for instance, I think one of the members here was interested in that. You might need to just keep aware of that. So yeah, I think this is a very good strength that you guys have a biobanks that can already be accessible for single cell sequencing.
Jay, I think,
yes, Masako?
Yeah, so Jay, you said there are some like the important part for like the having a biobank is how you store the samples, right? Does the Human Cell Atlas group provide how should we store the specimens from the after we collected the samples? I think that will be a very good information for like everybody who start collecting the samples.
Yes, so there are general approaches are kind of the snap frozen. So if you're taking resections or biopsies, I think the most standard way snap freezing. But once you do that, you're committing yourself to do nuclear extraction, right? Because it is nearly impossible to dissociate cells once they're frozen. So that's why I think, you know, biobanks that already have been collected, you may have to resort to nuclear sequencing. However, if you do plan to do whole cells, I believe there are various protocols out there to encourage people to dissociate first and then freeze down, and that preserves the quality of the RNA. And there are various protocols depending on the tissue and the cancer type that you would need to implement and test and verify and QC. And HCA does provide that either at the protocols.io or from Orit's presentation. The standardization working group also provides some recommendations. So I don't know if that answered your question.
Yeah, I think that's great because we do have biobanks, quite a few in the country, and some of them are very populated with completely different population characteristics. I think this is great, but many of them are also collecting prospectively. So it's very good to have this information about how we could move from now on if we want to include single cell analysis. Yeah, that's really great.
There's a spatial technology that are on the horizon as well. So if you have biobank samples, you can already do spatial transcriptomics as long as the RNA is good quality.
I think it's also important to remember that a lot of these methods are still under development, and there isn't a perfect way to isolate all of the cells from any one tissue that's universal across all tissues. So you really will want to draw on your own expertise of working with that particular tissue type to get the best out of it. And if you're interested in the immune cell components, then particularly the myeloid cells, the granulocytes, are very hard to isolate using disassociation techniques. You may be better off with the snap frozen if you want to capture that immune component.
That's also great to know. Okay. I don't know how it works for other countries. We have Mexico, Uruguay, and Chile here, and also the US. In Brazil, we have ethical committees that regulate the use of samples. And it's often complicated for us to ship samples to be analyzed abroad, unless there is a very clear need for that to be done. Does anyone want to comment on that? Does anyone have experience with this kind of work? Or we could also discuss that each country can manipulate their own samples and share data. Yeah?
I can say, or I don't know if someone else wants to speak.
You can speak.
Okay. We have sent samples indeed to the Sanger Institute to be, we have sent paraffin blocks and DNA, just like that. And it wasn't difficult to send from Mexico. We hired this UPS company called Marken, and they handled everything. Basically, they told us, "Don't worry, we see this with the authorities," and they just came, collected the box, and took it. And in 2 days, it was there. We sent samples to Brazil to Patricia Severino at Inca, who's working with Mari, and those were frozen samples, and they also reached there in under 2 days with the same company. So it seems that, at least for us, sending samples outside is not a problem. People say that importing them back is a problem. So we haven't done that yet. But
for us, here's the opposite. We have no problem receiving them, but if we ship them, it's very complicated. It's the opposite.
I would have thought that should be the case in a way, because you're sending samples outside, is your resource. But it doesn't seem, at least in my experience so far, it doesn't seem to be the case. It's been easy, at least to Brazil and to the UK, to send samples. We don't know how easy it is to receive them, but yeah.
The reason I'm bringing this up is because I think it's always feasible, and each country is going to have their own regulations, right? So it can be more or less difficult. So I think that is okay. But the reason I'm bringing this up is because we seem to have a path to go before we can really process samples over here. And there is the funding issue. So if we can get funding from partners abroad to run, for example, wet lab experiments, and we could do data analysis, or part of the analysis being done abroad, like spatial analysis, for example, where we're going to be sending up slides, or nuclear sequencing if we have frozen samples, that could be an alternative. Does anyone want to talk about that? If, for example, we would like to send it to, I don't know if Edmund is here.
May I talk about that? Has his hand up?
Yeah.
I think I just might have a comment, because since we are about to set up this from scratch here, maybe it might be important to think about technologies that we can collect the data right on the bedside, let's say like that. Because 10x is great, but you have to have the equipment and move that around, which I don't think it's a good idea. And for really precious primary samples, freezing them down and then shipping them abroad, I don't think that's going to produce really good quality data. At least when I was in Boston, and I used it to generate data in the Harvard single cell core facility, they were very explicit about us giving them high quality viable cells. So the data is not going to be good, at least with the in-drop-seq type of technology. I don't know how robust this becomes the last year or so, because there's been 2 years that I don't generate any new data using any of the single cell technologies. But as far as I can tell, primary samples, they are gonna be precious, especially for rare tumors. And we should also think about a way to collect these samples on the site of collection. And for that, I use it, for example, SQL from Alex Shelek lab. I actually used that in a postdoc in his lab came by to our lab and prepared the samples with us.
Yeah,
and I think it's a good technology for these more precious samples.
Yeah. Sequel has to be established in the lab too, so it's possible. Of course, we could have a collaboration to do that. But and I think everybody agrees with that. If we're talking about live cells and for transcriptomics of live cells, you are completely correct. We are here considering, besides that, to have collaborators that could sequence your libraries, or that they could do spatial biology in slides, or single nucleus sequencing on frozen tissue. But you're completely right. It's very important for us to keep in mind that if we want to sequence live cells, it's going to have to be on site, or somebody can sequence your libraries later on.
Yep.
Yeah.
Just to keep in mind as we are about to design this. Sorry.
Yeah.
I saw Mari had her hand up as well.
Mari?
Yeah, I did, but actually you make the points I would like to make as well. But just to let you know, guys, that we are trying to acquire the 10x equipment, and so it would be easy also to manage the libraries preparation and so on. So we can.
I think we're gonna. Yeah, I think we already have Wilson with one machine in São Paulo. Lucio has one. There is another one in Ribeirão Preto, I guess. So we need many ones because we cannot be also walking around with the cells. So it's great to know that you're gonna have one. Okay. Anyone else wants to comment on this?
It's not directly related to this, but now that you're talking about reagents and all of that, I don't think this is highlighted enough in the HCA talks. Actually, when you are registered with the HCA, you get some discounts. I have just posted something. It's not too well formatted, sorry. You get some discount in 10x Genomics, in BioLegend, NanoString, and Takara Bio, and registering is free. I don't know the specifics about those discounts, so sorry about that, but you can email them and ask them about it.
That's great to know. Yeah, I don't think everybody here was aware of it, but yes.
Yeah. And I think, yeah, for setting it up, I think it's actually pretty important. And yeah, it's not to highlight it. I didn't even know about this.
It's really great to talk about that. Yes, it's true. I mean, everything that we can like cut costs on, it's really important. This could bring us to financial resources. We have seen here that quite a few groups have infrastructure to help out and could do part of the work, right? But there is having access to funding for the experiments themselves. Does anyone want to comment about that? Ideas on how we could fund groups to collaborate?
I'd like to talk a little bit about that. Of course, today, the current situation, I believe, in all Latin America is a little bit hard for funding. I believe all countries are dealing with the same issue. But of course, we can once, at least once we have the funding for at least generate an experiment, and we need to have, I don't know, money for that analysis. There are several initiatives that I when I was in Brazil, I never heard about, but here in Chile, I have seen there are several fellowships that you can send students abroad along South America and Latin America. For example, last year, a student was for 6 months in Cordoba, Argentina, with Elmer Fernandez. And this is a very interesting way on how to send some student to have funding to send some student to other groups, to establish the collaboration, also for the training of these students. And also there are some grants for, they are not for to fund your research, but at least to fund a collaborative initiative. CITED, there are several that every year we have these grants available, and I believe we should do something with that related specifically to single cell and in Latin America. Yeah? I believe this is something that we need to explore more.
that's a great point. We don't always have access to all that is available, and we tend to be looking always at the same agencies. That's very good. I think that if we can have a small group sharing these possibilities, it's great. And for sharing, for the transference of students, I mean, yeah, for training, that's what we need. Thank you. Yeah, let's take note of that. Daniela, yeah?
Yes, I'd like to share there as well. Of course, as I did say, I do not have experience with single cell sequencing yet, but I have seen that there's several international consortia or foundations that do award fellowships and grants to Latin American countries. For example, the Wellcome Trust has very good fellowships that will fund, for example, a mid-career researcher in Latin America for do your experiments. You don't really have to justify a lot what you want to do. Just have a a research plan, and you can use it as you see fit. That's a very good option. The Medical Research Council in the UK, if you have a partner in the UK, they will send a good good money and a part of a good grant to Latin America. And the Newton Fund, does a lot of funding also. In the last round was for Brazil. I think they have a country or 2 or 3 countries that they choose somehow every round. Okay. And the last one, as I remember, was for Brazil. So those ones are also very good options. That's for training, indeed, with a partner in the UK that if you have someone that you collaborate with, and they fund all the travel, all the conferences for them to give and teach, and you can go and work and that kind of thing.
Yeah, could we consider then that funding, even though for us it would be a challenge, we do have opportunities that are unexplored, and those opportunities could be considered strengths. Like in 2 minutes, we listed a lot of things that maybe are not within our radar. So we could propose to have a small group of us that are going to be monitoring and sharing this within Latin American interested groups.
Yes.
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 There are some considerations related to the funding from UK. For example, the Wellcome Trust funding. Chile and Uruguay, they cannot have access to that because they are
high-income country.
Yeah. For example, we are not part of officially part of Cabana, but we are unofficially part of Cabana.
But if we are all collaborating and someone gets the grant, then it would be okay, right? So I think the important thing is to be in touch so we can know about the opportunities. And
something really nice is that Chile started to be part of EMBU as a foreigner country and can apply to some funding from EMBU directly. For example, for the course that we are discussing with EBI, it would be an EMBU workshop.
That's really great. That's really great. So let's put it that we would like to have this little committee group of people that are going to be monitoring and sharing between the groups that later on decide to really be part of this and communicating, so we can all have access maybe to this. Okay. And it's great to know that Chile is doing so well.
So can I comment about
yes, sure.
So equity and equity working group was presented yesterday, and I think this is one of the major missions of the Human Cell Atlas, and to ensure that the data is actually generated by the sample providers themselves, and that it's not shipped across to Boston or to UK, but it is actually building grassroots research groups that are able to generate their own data set and deposit into the Human Cell Atlas. So there is a strong motivation, and I believe CZI and other funders who have been supporting HCA are also on board with that mission. So what I mean by that is if there's a push to create such programs where you train the people and provide the pipelines and the network as long as you are providing value to HCA database, that will have a good, I guess, an opportunity to apply for funding with CCI. I guess CCI usually is kind of, HCA is a healthy reference. So we talked about cancer previously, and it may be difficult just to say we will profile cancer samples. So you will need to consider how to balance the healthy aspect and the disease aspect when you're applying with CCI. But nevertheless, the motivations to make the data set more equitable is high priority for us.
But you know, one strategy might be to actually invite those funders to come to your planning meeting. Ask them to sit in and see where the priorities are for you and help them set the right scheme for you. And I think that you will find that the CZI, funders like the CZI, Wellcome, Gates, and so on will come and spend time talking to you about what your needs are.
I think that's a great idea for one of the ways to go. I think that's a very good idea. Get the funders together and see how they would like to see this going. And we would share with them what we have and what we're thinking and hear from them.
Yeah, I mean, I think it's important that you're helping them set their funding agenda.
Okay.
If this is going to be truly equitable access to these and equitable about capability building.
I think that's a great idea.
I also want to mention, you know, NIH has many funds that invites foreign collaborations, or even foreign researchers can apply directly. So there's one that
that's getting tougher, Jay.
It's getting tougher.
It's really tough.
But maybe finding a partner like Masako.
Yes, Masako.
and co-apply, right?
Yeah, I'm also happy to. Actually, Albert Einstein has the 10x machine, and like we have the sequencer as well. And like we are also the partner with the New York Genome Center. So like that's the New York Genome Center has like a lot of like the technical development as well. So like if you know like the starting collaboration is the good thing to like for like making like a good network group. And then I'm very interested to the work on the Hispanic Latino population in here, as well as the country. I should not say, but the original country as well.
Mm-hmm. That's a great idea. I think that is a great idea. Well, we're coming to the end of our session together.
Genetics.
Yes, Jay, please.
Oh, no, go ahead. I'm done.
No, well, yeah, no, it's just that we have some time, and I would like us to know if there is something else that everybody has the urge to talk about. We have some minutes still, so it would be good if we would like to discuss anything else. Okay,
well, I was going to mention, so genetic diversity being one of the importance. And so, as I mentioned in my presentation yesterday, even Asians are underrepresented significantly and makes the clinical use very limiting. So yeah, I think there are fundings as well that really encouraging more data production from underrepresented regions. So I think starting with genotyping and doing some single cell PBMCs, it's not a very sexy disease per se, but I think it really helps to learn the ropes, trying to get IRBs approved, the ethics, learning how to multiplex and building the communication channels across different laboratories, kind of starting with PBMCs and genotyping and trying to integrate data set and build upon from that experience to more complex tissues. And this, of course, you can start with cancer or start with brain tissues.
I think you're making a very good point, Jay, about how do we want to start? Because if we pull together to start with something that is maybe not that fancy, but everybody could collaborate a little bit, you know, and we could find a way to finance genetic diversity, which is something that hasn't really been.
May I comment?
Yes, of course.
Jay, you said to study genetic diversity for reference population or for disease. What are you? Because
I think it's reference population.
Yeah, because yesterday I mentioned in my presentation, we are maybe in October we are launching one project for sequence is at least 1,000 people from the Guarapuava city, and the idea is exactly to know the genetic diversity of the city and use this information to try to implement the precision medicine. And also we are, of course, collect samples from different diseases, including cancer. And maybe we can discuss more about that, because it's not the representative from Brazil population, but from the cities, specific from the Guarapuava city. I think it's more easy to know and to manage the information, try to understand one part of the diversity of the Brazilian population. Maybe I think it's very nice if we can discuss about that together.
Yeah, I think that's excellent, and if you already have the access to cohorts and individuals,
yeah,
only thing that you can do is then add the single cell PBMCs, right? So if you already have plans to do genotyping or genome sequencing, you already have the samples. So you just take the PBMCs and run, and you can do EQTLs, you can do very interesting additional analysis that could be very valuable. So yeah, I think that's a really good starting point, which may help to build connections. Yeah.
Guys, we have Enrique Hernandez who wants to ask a question too, or to contribute.
Yes, I wanted to contribute by telling that perhaps half of us are computational biologists, so we have a strong analytics, let's say, power here. And from a past experience with the International Cancer Genome Consortium, we had a lot of trouble in setting up like analytical standards of analysis. So perhaps the Broad Institute pipeline somehow established as the standard, but there were many different types of analytical approaches that were not like given a following. So perhaps we may think about forming some kind of network or some kind of task force to join forces and to establish the best practices of analysis for this type of data early on, so that the different expertises and so on may, in some sense, converge to have well-defined, high-quality computational pipeline. So that may be a good goal to start or to establish here.
That's a great point. Do you want to comment on this, Jay? Or Enrique?
Yeah, maybe Enrique can comment quickly, but maybe I would highly encourage you to connect with the global HCA. There are an analysis working group who have been investing a lot of time to set the right standards. However, I do recognize that it's not always easy to connect with the global HCA, and also a lot of these pipelines, depending on the technology you use and which biological questions you may have or which analysis you plan to do, So I agree. I think it's excellent idea to formulate a regional working group regarding the analysis, but at the same time trying to stay in line with the global HCA, so you don't necessarily reinvent the wheel. Yeah, but then still you have your own kind of ownership and leadership into that in the region. I think that's very important.
I think that's a very good point. I would suggest, I don't know what Vinicius, Wilson, and Eduardo, all the other ones that are working with data analysis, maybe the way to go would be for you guys to have a group that would join forces, sit together, and think about how to go about this. So people are not reinventing the wheel, or not like maybe even within Latin America redoing the same things. I don't know if you think this is a good idea. Eduardo wants to comment, please.
Hello. Yes, so I'm developing my lab what I'm calling a framework for unified single-cell analysis. I try to follow the standards from the AGSC as best as I can, but the software is still under development. And it's going to include features for multimodal single-cell analysis to integrate transcriptome, CiteSeq, and also single-cell ETAC-seq. And also, it's going to put together in the same computational platforms the algorithms that I have been developing over the years, including classical clustering, differential expression, but also trajectory inference algorithm that I have developed in the past year, and another ones that I have been implementing in my lab along the year. So I'm trying to put together a computational platform that integrates several features that are used for single-cell analysis that are not yet readily available through any other software package. So it's not going to reinvent any analytical tools that are already available, but it's going to include the new tools that hopefully new tools that I'm now developing in the lab to integrate single cell data in multiple levels in one single platform. So we can push together to maybe make this high standard in terms of the quality of the software, because I'm pretty much developing this with scientific initiation students, so it requires a lot of software engineering to make this globally useful, let's say like that. Thank you.
I think that's great to know. I think that's aligned with what we were just saying. There's a lot of competence on that, and you guys have to get together to discuss that. And Enrique from the DCP just shared here some pipelines already implemented by the HCA. I think that what you just said, Eduardo, is great. It's not about redoing what's already there. So I think that it would be great if you got together to check it out. Let me just tell you something here. One of our participants is suggesting that everybody writes down full names and emails at the chat so we can talk together after this. Christine, you want to say something?
I could see that Enrique had an additional comment.
Yes,
Go
I
ahead,
did. So I don't know if any of you have joined the HCA Slack, but it's open, and like adding to what Jay said, in the HCA Slack there is a data pipelines team channel in which you can chat with Kylie, which is kind of the manager that, and with many other people that are working on it. So if you have any questions, just drop in there. People is really nice, so feel free to ask any question in there, and they will be able to answer better than me.
That's great. I think that's great. I think we all have to keep in mind that the HCA during the past years, they have already come a long way on a lot of aspects that we are discussing here, even on biology protocols, data analysis protocols, even ethics. They have a lot of working groups dedicated to all these aspects. So we really have to dig into it, you know, and try to reach out and to collaborate directly with them, so we don't have to start from zero. So the idea is, since we're starting, let's start, like, take advantage of all that. What we want is to contribute with the HCA. So also, so I think it's important that we keep that in mind. It's a very good point, Enrique. Does anybody want to share any views on what they think HCA Latin America should look like? Yes. Is this all too new for us to imagine what a Latin American Human Cell Atlas would look like as a group?
Well, I think it would be important to highlight if there are any differences in, even if it's some of the same diseases or tissues that we study, you know, as the main Human Cell Atlas. I think this diversity should be one of the first points. Like, is there, I don't know the answer, I don't know enough about the literature in this topic, but is there any difference in gene regulation, you know, that is more prevalent in certain ethnicities that can be related to certain diseases that are more prevalent in certain places? For example, here in Mexico, of course, we're the most obese country in the world, and that's not only related to diet. Of course, a lot of that has to do with diet, but we also have one of the highest prevalences of certain polymorphisms that are, you know, just associated with obesity. So, for example, is there a difference in genetics, you know, at the level of gene regulation? Can you see different proportions of cells doing certain things than others in certain organs? I don't know how that would look like. And of course, focusing, I would think, in diseases that are important for these countries that, you know, maybe also important in other places, but sometimes have been just overlooked because they're not that common in other places. So I would think that's the best, at least in my point of view, view of how this could look like as a Latin American initiative.
Do we think that if we set a goal to have in 1 year some ideas of, for example, important cohorts of diseases which are most prevalent in our countries, or access to samples that could address genetic diversity, just like Wilson is constructing here? We are also going to have one on rare diseases. So Albert Einstein Hospital is sequencing thousands of genomes also, and we have over 1,000 also genomes that have just recently been published from Brazil. So I believe the other Latin American countries may have that. Do you think that in one year we can come up with that? Do you think this is a realistic goal that we can get together to organize this framework for HCA Latin America?
Why not? It's a great goal.
Yeah, that would be a more or less short-term, well prevalent diseases with cohorts, genomic data for genomic diversity.
I'm not sure about the genomic data, at least here though. I mean, we for sure can get probably together cohorts, you know, collaborating with hospitals that have access to these patients of, you know, from diseases with importance here, but I'm not sure in a year we can generate, at least.
No, I'm not aware. Yeah, I'm not sure about generating, but having a complete picture of what is available. So each, so like for us to design what we really can do, first we have to have access to groups that have it and have a clear picture of what we could do. So have this framework, and we can create a working group that would be in touch to collect this kind of information. And then from there, well, meanwhile, of course, we're going to have all these projects that we are going to try to get funding for, but maybe have this framework in a year timeline. Mauro wants to say something?
Yes, Patty, just a thought to share in the group. According to the data generated by the consortium. How is there a framework that we could understand and look at about how to share this data, for example, for data analysis, and maybe to organize working groups dedicated to understand and for example, to bring their methods to assess these data sets, these very interesting and very rich data sets?
I'm not sure I understand what you're talking about. Are you talking about data sets that already exist? The HCA ones?
I presume that the Atlas has generated single cell data that still
Enrique has shared here in the chat how to have access to pipelines, and also yesterday in the talk he shared how we can have access to human cell atlas data that is already available. I think you can reach out to Enrique to understand more how this works, but yes, most certainly, yes, they are already available. And that would be one of the task forces. Oh, Enrique is just sharing here again in the group of people who work with computational biology and data analysis, getting together to understand how these resources are accessible.
I actually ask this because this is one way to organize groups in terms of non-funding requirements. I have some experience with the CGA analysis working groups.
Yeah,
because they also produce a lot of data that need inputs from different expertise. And the way they organize groups is very interesting in the sense that they have clear targets, clear goals, and get together people that can contribute with their expertise. So I think I see that the Atlas has a very unique and rich data sets that can be shared by the community. And
I think that, Mauro, yes, that is a very good idea as one of the working groups. And there are quite a few of you guys already working with these data sets. Wilson, Helder is also doing this. I think Vinicius too. So I think, Mauro, it would be great if as one of our goals, data analysis is already happening when we are ready to generate biological data. So I think it's really important what you're saying, because this requires minimum funding at many times and can be already done. Can we also say then that we have a relatively short-term goal of this first year that data analysis groups can be organized into that? What do you think, Mauro and Wilson, Vinicius, and Hernandez from Mexico, Enrique Hernandez from Mexico?
I guess it's a doable goal.
Mm-hmm. At least setting yourselves as a working group.
Setting groups, it's yes, most likely a doable. Mm-hmm. And with all the previous experience and all the previous guidelines that we already have, we may even have not starting at zero, but having like wrapping up everything already established and trying to adapt it to the way we work in Latin America.
That's great. Guys, we unfortunately have to finish the discussion here because we have to, in 20 minutes, get together in the other room. Remember the other room where we were before, the webinar room, to communicate to the others some of the points that we were discussing here. So it was great talking to everyone here. We will get back to everyone, you know, like, we will have this, yesterday, this was mentioned, we're gonna mention it today again, that we're gonna try to have follow-up meetings, more informal in the next month, so that people can come back and reconnect so that we don't lose this, we don't miss the opportunity of really have this going now. So I think there's going to be 1 or 2 meetings opportunities per day the next month. So this is going to be shared with you. And in those meetings, we assumed that we were going to really be able to put together people who are really interested in have this going for the next year. And then we're going to have smaller working groups to try to make this, this happen. But we will be sharing this all with you guys later. So thank you very much. If you want to go grab a cup of coffee or something, I have now to talk to Christina and Daniela, right? So we can put together some notes.
Yes.
Thank you, guys. This was really great.
Thank you. Thank you very much.
Lovely to
Great
meet you all.
talk to everyone.
Thank you, guys.
Thank you very much.
Thank you.
Thank you, everyone.
Thank you.
Bye-bye.
Patricia, what a fantastic job you did chairing that. Well done. It was really great.
It's such a great crowd. It's such a great crowd, right? They're all great.
Such great discussions. And you pulled out the themes so beautifully. That was really great.
Thank you so much, everybody, for being here. It was a great crowd. That's why, right? They're all so good.
Bit quiet at the beginning, but
that's normal, right? Not everybody.
It's also very early at some place. Not well, I shouldn't say for me, but I do work at nights usually. So for me, it's like this is why I look like this. It's just like I woke up and so at the beginning it was a bit hard. But we're fine now.
It's true, I guess. And also not like some of the people here that were here, I know them, they're very chatty, but it's hard when you have like 30 people, right? To be able to like discuss, right?
No,
So,
you were great.
Interesting points here.
And yeah, so amazing actually how thematic it was. So Danielle, what were your main themes?
I don't know how we can I don't know how we can do this this feedback.
So should we write a thing in a Word document and read it?
That we could share? Yeah. Did you? I wrote it in a paper here. Did anybody already write it in a?
Yeah, mine is the same.
Yeah, I write in paper.
I wrote in the computer, but if.
So if you wrote in a computer, maybe you can read it to us and then we can maybe add. What do you think, Christine?
I really loved the way you framed the forward-looking piece. You know, this is our goal within a year. These are what the working groups can probably achieve. Why not let's frame the discussion around that? Because then we can talk about the challenges and opportunities about the opportunities with the biobanks and the challenges really understanding how to set the methods and how to generate the data in the various sites. The opportunities where you've got very, very cohesive interest in biology, but, you know, the challenge of being so geographically dispersed. And reasonably isolated pockets of people too. And then the opportunity of having so many people interested in the bioinformatics, the challenge of really understanding data interoperability and the importance of connecting back
and
to
also
the
to
HCA.
get them to talk together and define how to work, because they tend to be very within their walls when they have them.
I can tell.
Yeah, they all are already like developing as something else, right? And to get them down to earth to analyze data that needs to be analyzed is that's a challenge. But let's not point it out.
No, but you know, by setting your 1 year goal in a year, we will have reviewed how best to interact with the biobanks and set the themes that we're going to really focus on, and bring the funders together and have some further workshops to talk about how we network. That's a brilliant goal. And through that time, these data analysis people have been tasked with setting up some training workshops and working out how to operate with the data portal so that we can exchange data between teams.
Do you think we have to have a PowerPoint for that, or can we just say it?
I reckon you've got it.
I'm trying to organize because I wrote us.
Oh, that's great. We love that.
I'm trying to organize it in points. So we have points.
Let's say that, yeah, we have a 1-year goal of understanding how much information we have in our biobanks, cohorts, and the aim would be to work with genetic diversity or specific diseases. So we would have this time frame to understand that. We would also like to set up meetings with the funders, and that could be international ones like the CZI, maybe the NIH. I don't know how accessible they are, but the Gates.
They can say no. Why not invite them?
And welcome is amazing.
The welcome, yeah, the welcome, Gates, and in Brazil, the FAPs, they're called, the FAPs, for a discussion on what we have and how they would like us to frame our project proposals that would be competitive and interesting for the HCA.
How do you spell, sorry, these Brazilian?
It's FAP. It's Fundação de Amparo à Pesquisa, and they have one in each state of the federation.
Okay.
So I think that's a national one in Brazil that can fund collaborations, and many times they have international collaborations that they fund. So meetings with the funders, and then we would have working groups with the bioinformatics people, so they can already start training students and organizing themselves on how to work with us.
Yeah,
not with themselves.
And interoperability of the data with the HCA data portal.
Yes, that's very good too.
If they can't work it back with that, then it's going to be very difficult for you. The last training piece.
How they can operate with the HCA portal and all the information that is available.
And the last training piece that you drew together really nicely was all of these side groups, fellowships, exchange opportunities to bring, to help other incidental training that might sit outside the HCA, but allows for a lot of networking of people.
Yes, so we would have
I can't remember all of those different fellowships.
Yeah, we want to set up a group of people who would take care of that, exchanging.
Yeah. I'm just going to remove Joanna.
Okay.
I don't know. I think that's what is that? I don't know.
And maybe she's part of it. Do we have what we need?
Are you putting that in a PowerPoint, Daniela, for us with pictures?
Okay, I'm writing. Well, I'm writing a Word document organizing the thing, but we can. I mean, when it's actually organized, it's easy to put in slides.
Oh, okay, that's great.
Put it in different, because I wrote a lot of stuff here and it's from different places, so I'm trying to just put it. So I'm organizing it first, the goals. So yes, there's a 1-year goal, which is, as we were discussing, understanding what's available in different countries and having a complete picture of what we can work with and what we need. And then there's kind of side goals, I would say. I mean, you know, like other projects that we can pursue now in a way. So I don't know how to write that. Like,
yeah, you can write like immediate goal, we have a lot of common interests within the group. So you put goals, immediate goal. We have a lot of common interests, right? So we could get together in small hubs to
specific projects.
Yes, specific projects, right? So there is that. Then in that's immediate. Then in 1 year, where do we want to be in 1 year? So in 1 year, so we're going to be working together this 1 year to understand the biobanks, resources, specific diseases, so that in 1 year from now, we will really have a clear picture of how we can collaborate as HCA Latin America.
Jay was suggesting an immediate goal to you should be just to choose some simple tissues and benchmark between the sites how each laboratory, basically being confident that each other is generating equivalent data with equivalent cells.
Can this go together with the bioinformatics working group that would be getting together to?
I think this would be the data generators, so the people who have the machines. Yeah.
Ah, okay. Oh, yes. Okay, that could be one of the immediate goals, right? People who have the machine. Yeah. So specific projects and something like that. Choose a kind of sample that would be more genetic, just like PBMCs, right? And start working.
He was suggesting PBMCs, and what they did in Japan. And what we did in Australia was have some blood that was taken from one set of donors, shipped between the different sites.
Yes.
And then everyone did the library preparation at those sites. And we just came back and compared data and said, so if we get data from Mexico and we get data from Brazil, is it the same? It's a very interesting benchmarking activity, but it does take a lot of trust, but it lets you know whether or not the methods you've got are robust.
Okay. Yes, I think that's okay. Good. So those would be two immediate things: setting up small projects and getting people who have the machines to work with common samples.
Just to work with each other, yeah.
Yeah. To be able to work with each other and with the protocols, right? Immediate also would be the working group for data analysis that we were just mentioning. So workshops, learning how to work with HCA data, with the protocol, establishing, looking at the protocols that are already available.
Yeah, look, the protocols you need are so tissue specific that, my strong advice to you is you are the experts in the tissues that you're working with. You're going to be able to work out the best way of isolating that tissue. You can start with the HCA protocol, but a lot of the tissues you're interested in, like melanoma, are not in the atlas.
It's true. I remember Orit mentioned some of that too, right? That there are some specifics, and you try the protocols, maybe that's not really going to work as well.
Yeah. The blood, and so the only reason you would use something like PBMCs is so that you've got a benchmark to compare different facilities with. Because you're not going to use those protocols on a more complex tissue.
Of course, yeah. You'd be more like to learn how to work together, get used to generating the data, sharing the data, analyzing the data.
This is a reasonably cheap sample to get. How easy is it to ship between sites? Is it worth shipping, or do we lose the sample? Yeah.
Okay. And then also within this year, Daniela, would be the meetings with the funders.
Immediate goal.
Yeah.
It may take you a year to get them at the table, but
yes, within a year we want to do that. Immediate goal is the initial specific projects and benchmarking protocols and bioinformatics. Within 1 year, biodiversity, biobanks, diseases, and the meeting with the funders.
Yeah.
Okay.
And then there was the list, having a working group who was going to get a list of the training opportunities, the small grant training opportunities, which may be particularly facilitating laboratory exchange. So you can get trainees to come back with the EBI methods or the Recon methods.
Okay, coming. So,
they want us to be back in 5 minutes.
Okay, coming. I'm almost done. Okay.
I think you have to send this to me by email. Let me see.
Yes.
I'm gonna write my email here.
It's just that I'm trying to put. So very quickly, okay. There's the discounts and all these things.
Well, you can save the chat. Do you want to save?
Yeah. Think right. So I have here. Yeah. So I have here. I can send it to you. I didn't. Okay. Yeah. All right. So let me send you both. Oh, I can send it in the chat as well, right?
No, in the chat. I don't know how to pick it up from here, but maybe, yeah. I don't know. Do I download it?
I think you can. So I can send it both.
Yeah, just send it to my email. I'll get it there.
Okay. So I have it here. So email. Right.
It was great help, Daniela.
Well, I hear this. So is it the Einstein.br?
No, send me to this. I just put it here in the chat.
Morrigan PS?
Yeah, because the Gmail is faster. The lab one takes a while to.
Christine, do you want me to send them to you as well?
Yeah.
That would be great. Thank you so much. I'll send you my email as well.
I did not include here the participants' summaries, but I have them as well. So I can. I can put them, but they would be raw. I did not pull. I did not pull. How to say? Clean those ones, but I can add them at the end.
I'm not sure.
Okay. Yeah. So sending now. Where is your email? Yeah. Okay. Right. So it's gone. You should get them now, I hope.
Yeah,
so basically, while they arrive, I'll open it again. So I wrote down basically goals. Immediate goal, formation of small hubs to pursue specific projects. Immediate goal, choose some simple tissues, PBMCs from the same donors, for example, to benchmark techniques and results against the main HCA. Immediate goal, create working group for bioinformatics to learn to work with HCA data and learning protocols. Another immediate goal is meeting with funders and see what they're interested in to help them fulfill their funding agenda. And I wrote welcome CZI Brazilian FAP. And then 1 year goal, understanding what's available in the different countries, biobanks, resources, specific diseases, and have a clear picture of how we can collaborate as HCA Latin America. Then strength, lots of common interests, potential for collaboration, several groups in different countries with data. I wrote data, significant data analysis experience. Data analysis experience.
That's great.
Several groups with access to biobanks from genetically
diverse. Girls, I have to go because we have to be in another room.
Okay.
Well,
and if I don't leave this room, I cannot go in the other room. But we can communicate afterwards in the meeting. It was
Okay,
great.
see you
Thank
there.
you so much, Daniela. Thank you so much,
everyone.
Christine.
See you there.
It was a pleasure.
So we go back to the meeting,
Bye.
right? All of us.
Yeah, yeah.
Okay,
Yeah.
perfect.
Bye.
Awesome.